We're now in the second part of our non-fermenting gram-negative rod lecture. And in this lecture, we're going to talk about several other organisms. And the first genus that we'll talk about is Burkholderia. There are several different species within the Burkholderia genus. And the one that you would commonly see in the clinical setting is Burkholderia cepacia, or there's actually a whole group of organisms that are in that are members of the Burkholderia cepacia complex. But instead of speciating all of these different Burkholderia, since they're all very similar and they cause similar infections, they tend to all be grouped with Burkholderia cepacia. So the cepacia complex are nosocomial pathogens. They are associated with causing pneumonia, especially in cystic fibrosis patients. So again, we have our organisms that infect cystic fibrosis patients, our Pseudomonas aeruginosa, our Stenotrophomonas, and now our Burkholderia cepacia complex. Also, these organisms can cause endocarditis, urinary tract infections, and wound infections. They've been isolated from irrigation fluids, and anesthetics, nebulizers, detergents, and disinfectants. These are highly resistant organisms, and they are ubiquitous. So again, just like with Stenotrophomonas, you have to be careful and always check your disinfectants. So in the clinical laboratory, if you're using 1 to 10 bleach to wipe down the countertops and disinfect the countertops, you have to make a fresh solution every single day, or you have to use a purchase type of solution like a hype wipe that has an expiration date. Other types of detergents and disinfectants that may not have an expiration date, you really want to check them periodically. And if they need to be made every day or every other day, you have to be very diligent about doing that, because they can get contaminated with organisms like Burkholderia. The Burkholderia organisms will grow well on most lab media. However, after a few, like 3 to 4 days of growth on sheep blood agar, they will lose their viability. They will die off. So if you put Burkholderia on sheep blood agar and let it sit in the incubator for a few days and maybe take it out and come back several days later thinking you're going to do another test, you may not want to do that because it might not be viable. They will grow on McConkey agar. They produce a very weak, slow oxidase reaction, so they are considered oxidase positive, which differentiates them from the Enterobacteriaceae, but it's a very weak positive, so they can be difficult to do an oxidase reaction on. And they are oxidizers of glucose. So what does it mean to oxidize glucose? It means in an OF media, they will be yellow or acid in an open tube only, but in the mineral overlaid closed tube, the media will not change color. So this slide shows you Burkholderia cepacia growing on some selective media. So on the left-hand side, this is a agar plate that has a similar type of OF media in it. In Burkholderia cepacia, since it is oxidative, it will turn yellow in that media. There are other Burkholderia species. Burkholderia gladioli is a plant pathogen, but it does resemble Burkholderia cepacia. And there's also Burkholderia mallei. Burkholderia mallei is the cause of Glander's disease, which is a zoonotic infection, and it usually is a disease that affects livestock, typically horses. But humans can get infected with Burkholderia mallei. It's a very, very rare disease, so you really shouldn't be seeing Burkholderia mallei in the clinical setting, unless you're working in an area where there may be occasional infections due to close contact with livestock or horses, or if there is a bioterror attack, because Burkholderia mallei is a bioterror agent. Burkholderia pseudomallei causes the disease melioidosis, which is a very aggressive granulomatous pulmonary disease. So both Burkholderia mallei and Burkholderia pseudomallei can cause a respiratory disease in humans. Both Burkholderia mallei and Burkholderia pseudomallei are bioterror agents. They're on the select agent list, and these are actually two organisms that I just recently worked with, because I previously worked for the National Biodefense Analysis and Countermeasures Center, so I do know a lot about the Burkholderia organisms. Now, you can also get infected with Burkholderia pseudomallei through ingestion or through inhalation or inoculation. What you get is a metastatic abscess formation within the lungs. It gets into the bloodstream, causes an overwhelming septicemia, and that's what usually patients will die from with Burkholderia pseudomallei, is this septicemia. Pneumonia is a very common presentation in individuals that are infected. You can get local infections, such as a cellulitis. You can get draining abscesses, and there's a very long incubation period from the time of infection to symptoms. Now, Burkholderia pseudomallei has areas of the world where it's endemic. So parts of Australia, Mexico, and Southeast Asia, there are areas where you will see a lot of Burkholderia pseudomallei infections. In the United States, we do not see Burkholderia pseudomallei. So again, in a clinical microbiology laboratory in the United States, you should not really be coming across any Burkholderia pseudomallei. You will come across a ton of Burkholderia cepacia complex organisms, but you will not come across Burkholderia mallei or Burkholderia pseudomallei unless there were some sort of bioterror incident. Now here's Burkholderia pseudomallei on sheep blood agar. The organisms tend to be very wrinkled, kind of dry looking organisms. Now there's another family of organisms that fall into the non-fermenting gram-negative rods, and these are the Flavobacteriaceae family, and the genera within this family include Flavobacterium, Cresiobacterium, Meroides, Weixella. There's a lot of different genera within this family, and a lot of these genera used to be other organisms, other genera, and they've recently changed their name. All of the Flavobacteriaceae family members are ubiquitous in soil and water. They are not part of the human normal flora. Again, these organisms are associated with nosocomial infections. They are very, very weakly fermentative, so they are still considered non-fermentative. So they do look like non-fermenters, but a few of these organisms can be weakly fermentative. So the most commonly isolated organism out of the Flavobacteriaceae family is the organism Cresiobacterium enologenes, but this used to be Flavobacterium, the genus, and it recently changed to Cresiobacterium. The Flavobacteriaceae family member that causes the most disease is Elizabethingia and meningosepticum. Now, these Flavobacteriaceae members can cause meningitis or septicemia in premature newborns. They are non-motile. They tend to produce a yellow pigment. They can cause a lavender green discoloration on blood-containing media like your 5% sheep blood agar. They're positive for DNase, oxidase, gelatin hydrolysis, and weakly positive for indole. So they are oxidase positive organisms, so you can differentiate them from the Enterobacteriaceae family members. Moraxella and oligella are very strongly oxidase positive, non-motile, very tiny rods to rods. They are biochemically inert, meaning on OF media they will not change the media will stay green. It won't become acid in either the open or the closed tubes. These organisms are strict aerobes, and they are opportunists that can reside on mucous membranes. And they usually are susceptible to antibiotics such as penicillin, unlike many of the other non-fermenting gram-negative rods, which tend to be more resistant to antimicrobial agents. Now, we already talked about Moraxella catarrhalis in our gram-negative coccobacilli lecture earlier in the semester. So Moraxella catarrhalis causes respiratory tract infections and ear infections, and it resembles Neisseria. So we talked about Neisseria previously, gram-negative Diplococcus, and Moraxella catarrhalis is also gram-negative Diplococcus. Gram-negative Diplococcus, so that's why we talked about Moraxella catarrhalis with Neisseria. But there are other species of Moraxella that we didn't really discuss. So there's Moraxella non-liquefaciens, which is the most common Moraxella isolated. It resides as normal flora in the respiratory tract, and it does not grow on MacConkey agar. Moraxella lacunata is a common cause of conjunctivitis or eye infections. There's Moraxella osloensis, which is normal flora of the genital urinary tract, and Moraxella atlantae, which can grow on MacConkey agar, unlike most other Moraxella species. Oligella, the species are Erythralis and Eurylitica. These are small, paired, gram-negative rods or coccobacilli, very tiny rods. They are usually isolated from the urinary tract. They are non-oxidative, meaning that they will not turn OF media yellow. They will not produce acid. They do not grow on MacConkey like most of the Moraxellas. Shewanella, we have Shewanella putrefaciens and algae. These are rare pathogens. They have been isolated from abscesses and traumatic ulcers. They produce high levels of hydrogen sulfide on triple sugar iron agar. So these are non-fermenters. So they would be alkaline over alkaline on triple sugar iron agar. However, they would turn the triple sugar iron agar media black because of their hydrogen sulfide production. Shewanella algae is halophilic, meaning it's salt loving. It will grow in salt, whereas most organisms don't really like salt. We talked about our vibrios that some of them do like salt, and it's oxidase positive. And then there's our list of our less common non-fermenting gram-negative rods. We're not going to talk in detail about all of these organisms. It's just important to be able to recognize if you saw this organism in a list that is non-fermenting. These are not organisms you're going to isolate very often in a clinical lab. They have been known to cause infection. You can see them. And if you want more detail about them, you should read your chapter 21 in your textbook. Again, more or less common non-fermenting gram-negative rods. So in summary, all non-fermenting gram-negative rods fail to ferment carbohydrates. They do not ferment sugars. Most of these organisms are found in the environment. They're ubiquitous. They tend to prefer aerobic environments. Most of these organisms are opportunistic organisms that cause nosocomial infections in hospitalized patients, especially those that are immunocompromised. Of all gram-negative rods that are isolated in a clinical setting, approximately 30% of them will be non-fermenters. The rest would be your fermenting gram-negative rods like your Enterobacteriaceae, Vibrio, or Aeromonas organisms. Your Enterobacteriaceae are going to predominate. The most commonly isolated non-fermenting gram-negative rod are Pseudomonas aeruginosa, Acinetobacter baumannii, and Stenotrophomonas multifida. They're less likely to cause disease than some of the fermentative gram-negative rods. However, they are very highly resistant to antimicrobial agents, so they tend to be very difficult to treat, especially since they're opportunistic organisms. They cause infections in patients that are already very sick, and then they tend to be resistant to antibiotics, so these can cause some very severe infections. Laboratory diagnosis. Of course, these are gram-negative organisms. Stenotrophomonas multifida tends to be a rod. Acinetobacter tends to be a very short rod or coccobacillus. And Moraxella tend to be more cocci to coccobacilli, very tiny rods that you can barely tell are rods. Most of the Moraxella species, except for one, will not grow on MacConkey agar, but most of the other non-fermenting gram-negative rods will grow on MacConkey agar. Most of these organisms tend to be oxidase positive organisms. However, we do have some oxidase negative organisms, so you have to be very careful about differentiating some of these organisms from your Enterobacteriaceae family members. So your Stenotrophomonas multifida is oxidase negative, but it's an oxidizer. Acinetobacter baumannii, again, is an oxidizer, and Acinetobacter lawafii is non-saccharolytic or biochemically inert. We have a lot of oxidase positive organisms, and out of all of those, the Shewanella putrefaciens is an oxidizer that produces a high amount of hydrogen sulfide. We also have some pigmented non-fermentative gram-negative rods. So we talked about pseudomonids and how some of them form pigments, such as the fluorescence group, the pyoverdin, and the Pseudomonas aeruginosa that produces pyocyanin. But we have another group that can form pigments. So these are various organisms that are non-fermentative gram-negative rods that can produce various types of pigments. And these include Chrysobacterium meningosepticum, Flavimonas, Agrobacterium, Roseomonas, and Methylobacterium. So here is Chrysobacterium meningosepticum, and you can see that there's a yellow pigment when the organism is grown on sheep blood agar, but no growth on MacConkey agar. Achromobacter is another type of pigmented organism, and it produces kind of a pinkish hue on sheep blood agar. And Roseomonas produces a dark pink to red colony on sheep blood agar. Sphingomonas is bright yellow on sheep blood agar. And we are now going to move on to the third part of this lecture.